name is Isabel Ayora, and I'm a fourth-year student of Psychology and Genetics. I'd like to thank my supervisors, Mick Watt and Megan Wilson, and members of the Wilson Lab for their guidance. I'd also like to offer my immense gratitude to Susan Sim of the Otago Medical Research Foundation for funding this 10-week project, creating the opportunity for me to contribute to our understanding of cognition and the effects of stress on young minds.
1 in 6 New Zealanders experiences a mood disorder, such as depression. These disorders often include cognitive difficulties with memory and attention. How does this relate to adolescent, socially isolated rats? A healthy memory and attention both rely on having enough of the neurotransmitter dopamine in a region of the brain called the prefrontal cortex. To make these cognitive processes work, brain cells inside the prefrontal cortex send dopamine to each other. A special protein called dopamine transporter, DAT, keeps the level of dopamine in balance by clearing away excess dopamine in between brain cells. This prevents too much dopamine signal from happening. Dopamine transporter is encoded in a gene called DAT1. We predicted that normally the control area of DAT1 has a chemical tag called DNA methylation, which stops too much DAT1 from being made. But stressful conditions may cause this tag to be removed from the control area of DAT1, giving a go-ahead to the production of more and more dopamine transporter. Once the stress ends, normal amounts of dopamine may start being released, but the overwhelming army of dopamine transporter endures, so too much dopamine is cleared away. Dopamine then decreases below a healthy level, leading to difficulties with mood and cognition throughout adulthood. Our project expects the socially isolated rats to have less DNA methylation of DAT1 and more dopamine transporter, even after growing up and being reunited with family. While the paired rats are expected to have more DNA methylation tags on DAT1 and normal levels of dopamine transporter. At this stage of the project, with 7 weeks completed, we are still going right on schedule. The brain tissue has been dissected, RNA has been extracted and measured for DAT1 levels, and samples have been prepared for the detection of those DNA methylation tags. This week should wrap up the final data collection, with data analysis and conclusions to follow next week. Stay tuned for the final report.